Hello, can everybody hear me? Hi, so I'm Adriana. I'm a postdoc at the Transylvanian Institute of Neuroscience here in Cluj, and I will be one of the moderators for today. So welcome everybody, and I'm really happy to see everybody here, so many people from all kinds of fields. What I wanted to tell you a little bit about this event. So this is fueled by the collaboration between the Babeș-Bolyai University and Transylvanian Institute of Neuroscience. And it aims to bring neuroscience closer to the academic and the industrial environment in Cluj-Napoca. We want to make many meetings of this type, and all of them aim to bring excellent neuroscience researchers here at your disposal, so that you can ask questions and they can spike your curiosity, and then you can satisfy it by asking as many questions and participating as much as you can in this discussion. So I think I can tell you a little bit about the format. We will try to keep it as interesting and as fun as possible, so we will start by a short introduction by our organizers, and then we will go on to have 3 short talks by our speakers, and in the end, the most important part will be a discussion. And this discussion depends very strongly on your input, like I said, so please ask questions, give us your comments, your feedback, and try to get all of us to get as much as we can out of this meeting today. And I think that's it. I will try to introduce our organizers now. There's Florin Albeanu, who is a PI at Cold Spring Harbor in the USA. There's Andrei Miu, who is the director and founder of the Cognitive Neurosciences Laboratory here in Babeș-Bolyai University, and Raul Mureșan, the president of the Transylvanian Institute of Neuroscience. So, enjoy.
Thank you, Adriana. So we'll start now by introducing the speakers, and then there will be the presentations. I would like to introduce Tom Mrsic-Flogel, who's right now the director of the Sainsbury Wellcome Center in London. His professional career started with a PhD at Oxford in neuroscience and biology. Then he moved on to the Max Planck Institute for Neurobiology in Martinsried in Germany, where he studied with Tobias Bonhoeffer. From there, he went on to UCL again, to London, and then he went to Basel, where he established his lab at the Biocentrum. Later, he was recruited by the SWC as the director of the center, and his lab is doing very modern experiments in trying to decipher how neural circuits contribute to perception, and more recently, how these neural circuits are involved in decision-making and in producing behavior. So thank you.
It is my honor to introduce Professor Florian Engert. He's a professor of molecular and cellular biology at Harvard University. And if I were to say just one brief idea about what he does, is that he's asking the big questions about the brain in small brains, in zebrafish, actually larval zebrafish brains. So in this sense, I think he's one of the bravest and wisest men I had the pleasure of meeting, and I'm looking forward to hearing his talk.
It is my pleasure to introduce our last speaker, Georg Keller. Georg is a professor at the FMI in Basel, and his lab is focused on testing a very interesting idea that our perception of the world emerges as an interplay between sensory information that we receive from the environment and some predictions that the brain is making. And over the past few years, his lab has designed very elegant experiments and tested and showed that indeed our brain makes strong predictions and computes errors between what it predicts and what actually it gets from the environment. And as a result, it relies on internal models of the world that then is using such prediction signals to update such models. And such work has some profound implications for understanding how our mind works and how we actually construct a sense of reality from our biases. In addition, he's an extremely humble person and very, very shy, as you'll see. I think, Tom, it's your turn.
So thank you for inviting me, and it's a great pleasure to be in Cluj. It's a beautiful city. I was born in Zagreb, and I see many similar architectural features between Cluj and Zagreb, so it feels like I'm partly at home here. So we were really delighted when Raul and Florian invited us to give a talk here. And we discussed and bickered and argued about what we should talk about, because we were told it's a general audience. Normally we speak to other brain researchers, and this time we're speaking to the general public. So we tried to bring alive somehow the neuroscience research. I drew the short straw. What that means is I'm responsible in this first talk, not to talk about my own research, but to talk about generally a little introduction into the brain and how we study the brain. And then the more interesting talks will be given by Florian and Georg. So forgive me, but it's not my fault, okay? So I'll start with this. So there is an echo here. Is that a problem? Okay. So the question we often ask ourselves and often debate is, will we ever understand how the brain works? And then you ask yourself the question, well, what does that even mean? And I think that will cover in the later talks. So right now, my talk will be divided very briefly in what the brain does, and this is just to give a couple of snapshots. We'll hear more about that later. I'm going to briefly describe a little bit of what we know about the brain at a very simple level, such that we get everyone on board. And then I'm going to introduce some of the methods that will hopefully get us closer to understanding actually how the brain does what it does, okay? So what does the brain do? Just to get it out there, all your emotions, feelings, intentions, plans, troubles that you feel in your mind come from the brain. There's no other part of the body that generates this stuff. It's all the product of activity of lots of neurons inside your heads. So how you see the world, how you feel about the world, how you perceive the world. All of it, okay? And it's an amazing machine. But at the same time, part of that amazing machine can play tricks on us. So for example, if you can see this image, it might be difficult, but you see three people here. And I think that most of you might perceive the person here at the far end of the street to be bigger than the person near us. Does everyone see that perhaps? But if you look very carefully and you remove this context of the image where there's a perspective, those individuals are identical in terms of how much visual field they occupy, okay? So what this means is that the same visual input, the same person of the same size reaching your eyes can look very different in your mind. You can perceive it very differently simply based on the context. We can discuss later why the brain does that and how it fools you in this way. So this is one of the visual illusions that tells you that what is out there doesn't necessarily generate what's inside your mind in terms of your thoughts or perceptions. Now I'm going to show you, now I'm going to do a little test. So another thing, the amazing thing that certainly human brains can do, but also of other animals, is that it can learn. And it can learn rapidly. And this is an experiment using you as the guinea pigs to see how quickly you can see. So first of all, and I would like you not to shout out the answer if you know it, is I would like you to look at this picture, okay? And try and see what's inside that picture. I expect that maybe a very small number of you will recognize what is in this picture. So keep looking. Now I will say there is a frog in this picture. So can you raise your hands? How many people can see a frog? Can you raise your hand? Okay, so that's not very many. That's about 10% maximum. So now, this is how quickly the brain can learn. Now I'm going to show you the picture of the frog. Okay, can you now see the frog? So can you raise your hand? How many people can see the frog now? Okay, so actually, not that many. No, kidding. The amazing thing about this, those of you who can still now see the frog, you cannot unsee it. There's no way you will not be able to see the frog anymore. So in the 1 second that I show you that image of the frog, you could now interpret this image. The image is exactly the same as before, but something changed in your mind to help you interpret it, right? So our perception is not just dominated by the input that's coming into your mind, but also what we know about the world, and that informs our perception. And we're going to discuss a lot of that afterwards. So what is a brain? I think you all know what a brain is. It sits inside your head. Depending on the species, it has lots of, the number of neurons vary, the sizes of neurons vary. The key element of the brain is the neuron, the brain cell. And neurons communicate with each other through synapses. So this is what we call the cell body. These little processes are called dendrites because they look like trees. And then they send cables at the output of the neuron. Action potentials, impulses are sent along the cable, and they activate downstream neurons, and that's how neurons talk to each other. And the points of contact between neurons are called synapses. They're protoplasmic kiss, and this is what they look at close up. So on the side of the sender neuron, some chemicals are released, glutamate or other compounds, and that ends up to electrical change in the postsynaptic cell, in the cell downstream of it, and that's how impulses are transferred between neurons. There's many different types of neuron. Not all neurons are the same. They look very, very different, and the differences somehow reflect what they do in the brain. We will not really discuss that today, but there's a remarkable diversity that we already knew for more than 120 years or so. It is a highly complex network because you have lots of neurons. They all come in different flavors, shapes, and sizes. The human brain has 80 billion neurons, trillions of connections. It's the most complex organ in the universe that we know, at least. And it's a real challenge to figure out how it works. It's a highly complex dynamical system. Neurons, unlike in this picture, are much more densely packed. So they more look like this. And it's actually a very dense network. Unfortunately, the image here you can't see. This is really how the neurons are packed inside the brain. And this is just in a small chunk of the brain. And then neurons in a small region of the brain also can communicate with other regions that are much further apart. So for example, a neuron in the front of my brain here can send long-range projections to other parts of the brain too. The longer such projection is that in the blue whale, where the neurons from the motor cortex that controls movement goes all the way down to the spinal cord, and it can be 10 meters long, just a single cable of a single cell. Right? Okay, so just briefly, so it's a complex tissue, a black box that we're trying to understand. So how are we trying to understand it, and what are the methods that we use? So it started, even Egyptians started studying the brain. There was brain damage in certain soldiers injured in battle, and they figured out that they had certain deficit. But the modern day neuroscience started surprisingly with an Austrian, anatomist psychologist who was called Franz Josef Gall, and he used a very ingenious method that, based on the bumps on the skull, so nothing to do with the brain, he could try and figure out where different functions are localized in the brain. So if you look carefully here, this part is called cautiousness, ideality, hope, veneration, firmness, self-esteem, and so on. And he was just making this stuff up. Okay. But then came a very obvious study. As medics, doctors started studying patients that had certain brain lesions, they started realizing that there's a correlation between which part of the brain is damaged and what kind of behavioral deficit the person had. Okay. And with that, they start slowly mapping the function of the brain. I'm going to just give you a few examples here. So this is a very famous, one of the first detailed description of a person who had a very specific lesion, so damage to the brain in the frontal poles of the cortex. He was a railroad engineer, and he was overseeing some railroad works in the US, and there was a dynamite explosion, and a metal rod shot through his skull into the air, but very cleanly took away one small chunk of its frontal brain. The doctor that saw him afterwards, the physician, said he was amazed how little behavioral impact this had on him. He seemed to be completely normal to him. But later, his friends and family started complaining that he became a very rude person. He was urinating and defecating in public. He was swearing at people, hitting people. His ability to do maths, to speak, everything else was kept intact. So it seemed like something about his personality has changed in terms of how he interacts in a social context, but nothing else was, everything else was spared. And since then, since those times in the kind of mid-1900s, all the way till now, because I wouldn't say luckily, we have many humans who have different bits of brain damage due to, for example, stroke or surgery. We now have a well-documented series of brain lesions in terms of what impact they have on the behavior. So I'm just going to give you a couple of examples here. So for example, if you take this part of the brain, which is a cortex called the parietal cortex, so it sits about here, if one of the hemispheres is damaged there, people exhibit something what's called spatial hemi-neglect. They simply fail to perceive what's in one part of the visual world. So they can see what's on the right-hand side, they can't see what's on the left-hand side, especially if the right hemisphere is damaged, okay? And so you ask these patients to draw a picture of a clock, and they will draw half a clock. Or you draw a house, draw half a house or half a flower, all right? So there's something really weird about this part of the brain. Their eyes are fine, they can see it. And we know that they can see things. If you ask such patients, you show them a picture of a square, like a piazza in Italy, and they say, describe what you see. They will describe the right-hand side. But then you ask them, if you stand on the other side of the square and then describe what you see, then they perfectly describe the left side of the square. So it's about how the brain maps the spatial world and just divides it into two halves. Okay, then if you damage another part of the brain here, what's called the inferotemporal cortex, the lower temporal cortex, somewhere here, then some patients, some individuals, fail to recognize faces. So they can't recognize a person. They can recognize the eyes and nose and ears, but they can't somehow put them together into a coherent picture of an individual. And that type of damage is called prosopagnosia. You don't recognize faces. And then there are other parts of the brain, I think, that you might know better from your own experiences, maybe of family or friends. When there's damage to the motor cortex, which is over here, okay, different parts of the body might be affected, where the person loses the ability to move somewhere on one side, typically. Face, arm, legs, and so on, right? And we know that from lesions. Okay, so this is kind of stuff that we can gain insight from lesions. Sorry, I'll skip this. But now modern methods are now enabling us to image the activity in the brain, so without waiting for people to have brain damage, and see what that correlates with in terms of what they're doing or seeing, okay? So we use these machines called the magnetic resonance imaging, and they measure blood flow that is correlated with neural activity, and then you can give various tasks for the individuals, so the subjects, to do in the scanner, and you can look at which part of the brain's become active, okay? So here are some examples, and I know I just heard today that you guys might be acquiring or potentially acquiring a new scanner here that enables to do exactly that, so that's very exciting. So I'm not sure how much you can see here. So this is the brain, and you can see that when individuals are seeing images, then this back part of the cortex lights up. That's called the visual cortex, and that's normal because we know that that part of the brain encodes what you're seeing. It's responsible for your sight, your ability to see. If you ask a person to do some finger tapping, like this, then this part of the brain lights up, and that's the part of the brain that controls movement, okay, in particular of fingers. And then if you ask somebody to do some mental arithmetic, you know, compute what is x squared minus 2x if x is 5, and then this kind of more frontal part of the brain lights up. So that's more the kind of cognitive, function. And then it's really interesting, you can also look at how the brain changes and how it differs between individuals if they are seeing the same stuff or hearing the same stuff. So here, the subjects were asked just listen to music. And then you see this temporal lobe active, so that's what we call the auditory cortex, that's responsible for you to hear things and to perceive sounds. And this is for individuals who are not musicians, who they themselves don't make music. And when you present the same kind of music to individuals who are musicians, then you see another part of the brain active, and this is a supplementary motor area that is responsible for planning your finger movements if you're a pianist. So pianists have an additional part of the brain active because they might be probably kind of thinking about how they play the music they're hearing, for example. Okay, so this is called functional magnetic resonance imaging. So that's a pretty cool method. And actually this method, I'm not sure you can see a werewolf here. There's also a part of the brain that is responsible for your fear perception or for how much fear you express. And that area is called the amygdala. So what happens is when you show images of scary things to people in the scanner, then this part of the brain lights up. And what's kind of cool is that there's this guy, Alex Hanold, you might have seen a documentary on Netflix about, he does this free climbing with no rope and climbs really scary, difficult, rock walls. Even though he's a little bit afraid, he's way less afraid than most other people. And there is very little activation in his amygdala, as though he doesn't experience much fear, at least. Okay, so that's what we can learn with scanning, and there's much more sophisticated experiments that are carried out right now. This was just a snapshot. Okay, but we can also record brain activity, not just with these methods that can measure regional activity. We can also record activity from individual neurons, from which we have many in the brain, and we can use different types of electrical recordings. So we can put EEG, these are the electrodes that you can put on the scalp, but for individual neurons, we need to use microelectrodes of different types. They can be metal or made of silicon, and you can record anywhere between dozens to hundreds, even thousands of neurons at the same time in different individuals, either humans or experimental animals that we use for research, monkeys, rodents, even fish. So yeah, and so just to give you a couple of examples, what we as a field have found over the many years that electrodes were poked in the brain. So if you go to this inferotemporal cortex, where I told you lesioning causes you to neglect faces, and you show different pictures. So this is recordings in the monkey. There's an electrode lowered into the inferotemporal cortex, and these little lines here are nerve impulses of a single brain cell recorded in that region. So when you show a picture of a monkey, this neuron becomes very active. When you show a picture of a human, it also becomes very active. So these are two faces, but then you remove aspects of the face, so it looks less face-like, then the neuron fires a bit less. If you show a smiley face, it fires a bit, but if you show a scrambled image of these things, it fires very little, okay? So these are kind of what's called face cells, and this has been reproduced with more elegant studies. This is just one of the first recordings that was done there. Okay, then there's cool neurons in the kind of inferotemporal cortex and maybe also the hippocampus, where there are potentially concept cells or cells that encode, that represent individuals or groups of individuals. So this is a very famous brain cell recorded that was called the Jennifer Aniston cell, or Jennifer Aniston neuron, where this is recordings from a human epilepsy patient. Electrode was lowered, and lots of pictures were shown of different things, you know, of spiders, of Eiffel Tower, of different people, including Jennifer Aniston. These are the pictures in the gray box. And whenever you showed a picture of Jennifer Aniston, the neuron was active. When you showed pictures of other things, especially objects, it was not very active. And then also when you said the words Jennifer Aniston without showing the picture, this brain cell was also active. So it wasn't just something that was encoding a picture of Jennifer Aniston, but just the general concept of Jennifer Aniston. It turns out that actually this neuron also responded to all the other actors in the TV series Friends. So it was actually a Friends cell rather than Jennifer Aniston cell. Other cells they found were Star Wars cells, where the same cell was responding to Luke Skywalker and Yoda. Right. So this is how we can record activity, and just to kind of, I have a few more slides. We can also, and this is maybe, we can do some magic where not just can we image the we can record the activity of brain cells, but we can also image their structure. I told you about the synapses. We believe that synapses are the substrate for learning in the brain, so it's crucial for us to understand how they change, how they strengthen and weaken. And over the last 20 years or so, we developed methods by which we can actually look at the individual synapses in the intact brain and how they change over time, okay? And the way we do this, we need two bits of cool technology. One is a 2-photon laser scanning microscope that is an advanced microscope that can image fluorescence inside the brain at very, very high resolution, and requires a very powerful laser. Such laser that if you look into it with the eye, it would fry your retina or burn a hole in the wall. But we can feed that into the brain and then image a fluorescently labeled neuron. Now, fluorescence in this neuron, neurons in the brain are not normally fluorescent. And the way we put fluorescence in this neuron is by taking a gene from a jellyfish that is fluorescent, modifying it slightly, and using various vectors to put that inside the brain. And then that labels the cell with this fluorescence, such that when you zoom in, you can see these are the individual synapses on the cell. We can use a similar approach to not just look at the structure of the cell, but also look at the activity of the cell. And now we modify the same protein, but make it sensitive to calcium. When brain cells are active, calcium goes up. And when this modified protein now becomes brighter when there's more calcium inside the cell, which means when the cell is active. And using the same microscope now, we can see these cells blinking, so they're flashing, and this is when each individual cell is active. And you can see a movie of that. So now we can really look into the brain and look at every single cell in a small volume of tissue, which we can't do with electrodes because we're going in blindly, right? So that's a cool method. Another advantage of this approach is that because we can see the cells, we can follow them over many days, even months, to see exactly how the activity in the brain changes when the animals do or learn different things. Okay. Good. And finally, last couple of slides. Now, to understand the brain, normally what you do is you record some brain activity and you ask the animals to do or see something, experience something, and you make a correlation between the two, And if you find that certain cells, when they see a face, they respond to faces, you say, oh, this is a face area of the brain. Or when people move, and brain cells are active when you move your arm, but not your leg, then you say, oh, this is the arm area of the brain that controls your arm movement, okay? But that's just correlation. And now correlation in science is important, but correlations could be spurious. They may not be real, right? This brain area may not actually be causing movement. It might just have a copy of that signal. So how do we ensure that we know which brain area does what? And so a new method arose, and this is if we can add activity to a given brain region and see what the animal does, or if we can take away activity from a particular brain region and see whether the animal can't do that anymore, that's a causative experiment. That tells us directly whether that area is important for a particular brain function. So in the last 15 years or so, there's been a whole suite of new tools by which we can precisely manipulate brain activity in very specific structures in the brain and in specific cell types in those structures in the brain. So we become very sensitive to manipulating brain activity. This approach is called optogenetics. And now, again, we take a protein that is light sensitive from some algae and bacteria that live in the sea and put them in animal brains using various vectors. So now, when we shine light onto those cells, the cells either become more active or, in a different construct, we can shut them off and they become silent. So when they would be active normally, we can now shut them off. And let me give you an example. And the way these experiments work, we typically use rodents for these experiments. So there are brain cells in here that express these optogenetic constructs. And then we implant a very thin light fiber that delivers light, especially onto those cells, so we can either activate them or shut them down, okay? And let me just give you a couple of examples of this. So this is the very first experiment this method was used. This is a movie that might start. Yes, so you can see a mouse here. The mouse is in a box, and it has a fiber that you can barely see here. And at some point, the fiber will turn blue, and as soon as the light is shone into the brain, the animal starts rotating in circles. So this is implanted in a part of the brain that controls movement on one side, so the animal is running around in circles. So we've added brain activity, and now the animal runs it. And when you turn it off, the animal starts behaving normally like nothing happened. That's pretty cool. Okay, here is a more extreme example. So in part of the brain that's called your hypothalamus, there are specific neurons that we can label that control aggression. So normally male mice like to fight with each other, and these neurons are important for fighting. So when you put this optogenetic construct in those neurons and activate them by shining a light, let's see what happens. This mouse doesn't have another mouse in the cage, so now the light comes on. Now the animal is looking at this rubber glove and starts attacking the glove because you've activated these aggression neurons in the hypothalamus of the brain. And this now tells us that these neurons are responsible for aggression, because if you activate another group of neurons, the animal would not attack the glove, okay? And that way you can start dissecting what different parts of the brains do by simply seeing what happens when you either turn them on or off. So it's a very powerful approach for understanding causality of what different parts of the brain do. And finally, we can use similar approaches, both optical or electrical, to help actually, in this case, animals walk again. So in animals that have spinal cord lesions, you can plane some activity patterns into the spinal cord, and that helps the animals restore their walking ability. And that's now also being piloted in humans, not just animals. But you need to do the basic research in the animals before you can even think about going to humans. Shall I stop now, or shall I do the modeling of the brain? Okay, let's just 2 more slides. Another approach to studying the brain is to model it, right? If you can build it, then you understand it. Some people think that, right? That's not necessarily the case. So for example, this is in the computer now. We can build a very complex deep network that can model, in this case, the visual system. How the visual system in the brain processes images such that you end up with face cells and object cells here at the top. And some of these models have been very successful at identifying. You show random pictures to this model, and the model correctly identifies whether this is a motorcycle or an Eiffel Tower or Jennifer Aniston, right? Great, problem solved. But as you will hear from Florian Engert, this may not be a whole thing, because the information in this artificial network flows from here, where the images are, say, in your eyes. It goes only one way, okay? And finally, such networks, these are called convolutional neural networks, deep networks, and so on, which are the basis of machine learning and artificial intelligence. They are becoming more sophisticated. They can learn more, cleverer things, up to the point where, Garry Kasparov was first disappointed when he lost his world championship chess match, and more recently, Google DeepMind developed this AlphaGo algorithm that can beat the best Korean player in Go, which was a feat that people thought was impossible to do, right? And there's some interesting nuggets that you can learn from this. The question is this the right approach at understanding how the brain generates all the wonderful things that it does? We don't know. The likely answer is it's going to be a parallel conversation between individuals who work in machine learning and artificial intelligence and the kind of experimental neuroscientists who do actual tests and experiments in humans to see and find the kind of algorithms for how the brain actually does it. And with this, I would like to stop and invite my dear friend Florian Engert to the podium.
Thank you, Tom. That was remarkable. Yes. And keep that in mind. One of the messages that you probably have learned from Tom is that the brain clearly plays a role in taking in information from the outside world and representing it. And what I'm trying to convince you today is that that view might be maybe not entirely wrong, but might be missing a larger point of what brains are doing. So that idea is the one of passive information processing, which is at the heart of machine learning and playing AlphaGo. So I'm going to talk first about an alternative view of the brain, namely an active processing machine that's trying to control the world and not passively process it. And more specifically, a computing organ that predicts the world and generates models of the world rather than passively representing it. The first part of my talk will be mostly about the human brain. I will play with all of your brains. We will look at optical illusions, and I'll try to convince you that most of the things that you think you are seeing, you are actually making up, your brain is making up. And then I'll spend 5 or 10 minutes to tell you about research we do in the larval zebrafish. All right? So that's the premises. The brain generates a model of the world. It does not passively process it, and it does so by predicting sensory input that's about to come or that is present. It's trying to simulate the world rather than process it. It will then compare this predicted sensory input to the actual input, calculate some differences, some mismatch, some error signals, some surprise signals, and it will use these mismatch signals that generate behavior based on these computations. So that is the idea that I'm trying to drive home in the next 5 to 10 minutes with you guys. Yes. I think Tom already gave you a few of these illusions at the beginning, but that's basically the main message I want you to take away from my presentation is the brain as an active processing machine, your brains, yes sir, that are trying to interpret the visual scene and to make sense of it rather than just passively processing it. So that's similar to what Tom showed you already with the frog. Those of you who can spot the dog, please raise your hands. That's a lot. So those of you who don't, I'll help you out, yes sir. But, so that was a poor demonstration, I grant you, yes, but we'll be moving on. Here we have another image, and the main point is that the square A and the square B look like they have different shades of gray. Yes, that's what you think. This looks dark gray, this looks light. I'm telling you they have exactly the same shade of gray. And what your brain is doing, it's making shit up here. Yeah? And it's doing that because your brains are thinking that there's a shadow here. So the light grayness here must be a consequence of the fact that this is actually much brighter than this because this is seemingly in the sun. If you put a mask over it, you'll see that they are actually the same. The difference in shades of gray is a construct of your brain desperately trying to make sense of this image. It comes up with the wrong example. Now this is a fun one. I want you all to stare at the center. You're all staring at the center spot. Don't move your eyes away. But what you should be doing, so I can tell you already what's happening in your brains. Your brains are getting accustomed now to motion in this case. So your brains are predicting motion, and they think that motion is normal and motion is just the way the world works. If you now still look at the screen and put your right hand on the armrest next to you, and now move your eyes away from the screen and look at your hand. Those of you who sat in the front maybe will see that your hand now starts to move weirdly, expand and bulge in weird ways, and that is because your brain is now translating this motion, subtracting that motion from your hand such that now your hand appears to move. This is another example where it's not a prediction about movement or color or shade. This is a prediction your brain is making about the shape of faces. In this particular case, the faces always point outwards. They're convex, they're not concave, they don't go inward. That is a mask that's rotating, and what you'll see when it's coming around, your brains are refusing to believe that this is a hollow mask. Yes? All of your brains insists that this is a face which needs to point outward. So in a way what's happening here is your brain is just switching the source of light. So now the sun is coming, well, let's wait till it comes around again. Yes? The shadow is on the right-hand side, so the light source is on the left. When the brain comes around, when the face comes around, the shadow was on the left. So what in a way, your mind has a choice how to interpret this. It can either think it's a hollow mask that's rotating, but since you have not experienced in your lifetime hollow masks, right? You have experienced thousands of faces, you will go with the alternative interpretation, namely that the light source is switching. Here it's on the left, and now when it's coming around, the light source is on the right. This is something your brains have experienced a lot. The light source is arbitrary in the room, and that's why you can't help it. This is again your brain making shit up, right? Trying desperately to construct something in the world which is not the case. Yes? Oh. Yeah. Many of you probably have encountered distress, yes, sir. And the question is, what's the color? And there's been tremendous fights and arguments. I've even seen relationships break apart over distress, yes, sir. So, yeah. So, who of you think it's golden and white? Who of you think it's black and blue? Well, you clearly are wrong. You're insane. You're crazy. You need to have your brains examined. I know this is golden and white, this brain. Yeah. But I'll get to that, yes, sir. I'll tell you how we can use this. Well, the real answer, of course, it's in between. This is just zooming into the dress, and it's sort of a whitish blue and a brownish yellow. Yeah? And the answer, I'll tell you, is hidden in the fact that your brain is desperately trying to assign a true color based on context. In this case, it's the background illumination. And here we have an example where we have a colored cube. In this case, it is seemingly under yellow, illumination here under blue. And if you look at these blue squares here and these yellow squares here, if I tell you they are the same color, they're both gray, you would tell me it's crazy. Yeah. So again, it's the blue here and the yellow here. They are actually the same color. And if I put a mask over it, then you think I'm pulling a quick one on you, right? That when I put the mask over, yeah, I mean, just look at this. Yeah. That I'm adding sort of gray here. And sort of to convince you that's not the case, I'm just moving it slowly. Yes. And to me, that's one of the most crazy illusions that I have experienced. Yes. Again, for those of you who still don't believe me, this and this is the same shade of gray. Your brain is telling you that this is actually blue and this is yellow, and that is because it's subtracting, in this case, the background illumination. So if it's gray and you're subtracting yellow, you get blue. Yeah? And here, if you take yellow and you're subtracting blue, you also get gray. Yeah? Now, to test whether the fights about the dress are maybe due to this misrepresentation of the background illumination, I did a little trick with the dress and with the cube. Yeah. What I did, I took fragments of the dress, I make them smaller. There is still a big debate, is it white or and yellow or black and blue? And it's obviously white and yellow, yes. And if you now superimpose it
and let me just go back and forth. So it's the same image. Yes. I'm not changing anything on this patch, but what you'll see here clearly, you can't help. Even I believe now that it's black and blue. Yes, sir. And even I, and those of you who insisted on black and blue will grant me that here, under blue illumination, it looks white and yellow. And the secret is, for the dress itself, that it depends really what you think, what your brain is filling in about the background illumination here. If you think it's a bluish fluorescent inside light, and we don't know this, we can't extract this from the photograph, then you think it's white and yellow. If you think that if your brain fills in information and thinks this dress was viewed in the outside warm yellow light of the sun, you think it's black and blue. So it is really context-dependent filling in again. So, what you see. I think I'll start with that. In the next slide, you will be shown the picture of an ancient vase with 9 dolphins. Scientific research showed that young children have no problem identifying the dolphins. On the other hand, adults' minds are rather corrupted, so they may have problems spotting 9 dolphins at first glance. I'm sure you're getting really excited now about what you're going to see. If you have trouble spotting dolphins in the first 6 seconds, then your mind is heavily corrupted and you're seriously obsessed with sex. So I'll switch the slides now, and then I want you to raise your hands the moment you spot a dolphin, okay? 4, 5, 6 seconds have passed. I don't see many hands here. I think the ones that are raising their hands are either lying or they are, yes, sir. Well, I'm a little bit worried now about you guys, yes, sir. I can't believe, yeah, but clearly we have dolphins here, yes. So here, there's one dolphin, no? Here's another dolphin, right? Dolphins, guys. I mean, this is really disappointing. Yeah. I mean, I'm seriously, I mean, yeah. Anyway, I've told you your brain is making stuff up, right? It's seeing things that are not really there, and maybe the last example was the best demonstration about that. I'll spend the last 5 minutes to tell you now about we are using these ideas in the larval zebrafish. Larval zebrafish is a tiny vertebrate animal. Well, this is not really playing very well, but what you'll see here is similar to Tom's image of a few neurons blinking in a mouse brain. These are all the neurons in the larval zebrafish brains that we can visualize with respect to their activity. So this brain is blinking now whenever the fish is thinking about something. Yes, sir. And we can see all of the neurons because the fish is small, it's translucent, it's like made out of glass. And we can use the same trick Tom explained about putting fluorescent indicators in all of its neurons and then using the same high-powered two-photon microscope with the immensely strong laser to visualize now activity throughout the brain. That is an amazing power, but the question is how do we use it now to try to understand the brain? And for that, we need to put the fish in, we need it to behave. Yeah. We need to have it interact with the environment and ask whether even the larval zebrafish's brain is making shit up. But the important part here is it needs to behave, it needs to interact with the environment, and the problem is in order to take these images, it needs to be paralyzed, yes, and can't move. That seems like a really horrible conundrum, right? How do you make a paralyzed animal move, behave, and interact with the world? The way we do that is we head fix it, we embed the larval zebrafish in jello, but the tail is free, and we can now record tail movement, swim movements, which we can interpret and we can extract what the fish wants to do. Yes, he wants to swim forward here, maybe the tail is inclined a little bit to the right, so he might be making a right-hand turn. Here's a shorter swim bout, yes sir. So that works, but it's still not paralyzed, it's still moving a lot, which interferes with the imaging. So we went one step further, and we suspended the fish, we paralyzed the muscles completely. Yeah? So he can't move at all, it's completely still. And then we use extracellular electrodes to record from the nerves that go down to the spinal cord and elicit swim movement. They're not successful in eliciting, the muscles are paralyzed, but the nerves driving the behavior are still intact. And if you record from them, we can get these neural recordings that really look like the swim movements. So we can use these neural recordings in this paralyzed animal to infer what the animal intends to do. And now we can do a trick. We can do this online and feed this information back into a virtual reality environment that surrounds the fish, such that whenever the fish tries to swim, the world moves accordingly. So we give him agency. We give the fish, the paralyzed animal, the ability to interact with the world now. And this is what this looks like. So here we have the animal which is suspended, and we'll move now the world underneath him when he starts to swim. And I know this is not showing very well, but here what you see from the view of the fish, how the world changes, how he's swimming. He is hanging out on this red island, and whenever he executes another fictive swim bout, he's crossing over, here maybe to another red island, and he's now swimming happily through this virtual reality without ever actually moving a muscle. But we are coupling the activity in its brain with the signals it sends to its muscles. We record that and feed it back into the visual feedback such that it can now, like a flight simulator, swim through this world. This is the trajectory of this swim, and it looks exactly like trajectories that a freely swimming animal would perform. So we think these fish, we don't even know whether they know that they're paralyzed or not. It's really the same aspect of in the Matrix. Does Keanu Reeves, does he know that he's actually just fictively interacting with the world, or is this all real? We did offer our fish a choice between the red and the blue pill. And here is a cover that was never actually published, where we see a larval zebrafish now embedded in the matrix. Finally, I'll end on now asking the question, is the fish also making things up about how the world behaves, about the world, like you and the dress? In this case, we ask when the fish moves. Does the fish, when it moves, does it have a prediction about how the world passes past him? So for a strong tail flick, he presumably expects strong movement. For a weak tail flick, the fish expects weak movement of the world. Yes. So that's the question we ask. And we can do this easily now because we are in control over how much feedback we give the fish. And what we find that under high gain conditions, meaning for every tail flick, the fish gets a lot of optic flow. So it's a super fish now, where we can equip the fish with superpowers, where any little tail flick leads to a large displacement of the world. And then we switch to low gain. Then we suddenly make the fish into a weakling fish, yes, where even for a strong tail flick, it gets only a weak feedback. And what you see, within a few seconds, it realizes that, yes, and upregulates its swim power, which you can see here directly. Yes, so we know now that they're aware of the consequences of their action, and they can correct it. Yes, if there's a mismatch between what the fish is expecting to see, given a certain motor action, and what it's actually saying, it realizes that and it corrects its behavior. If we switch back to high gain, then it downregulates it again very quickly. So what this tells us that our fish, not only are they like Keanu Reeves in the Matrix, they are also capable of using the superpower we lent them with efficiency and skill. And so we're looking for another superhero who could do that, and it's the Hulk. The Hulk, yes, who, when equipped with superpowers, can handle it somewhat effectively. And why we think the analogy is really apt is because, like our fish, which has this green fluorescent protein inside, the Hulk is also green. Right? Our fish and the Hulk also share the same color, so I think there's really nothing wrong anymore with the choice of superheroes that we've picked. With that, I'll end. I'll just tell you that not only can we now study the brain in this animal, that the whole brain, that interacts with the world, it also, this is a larval zebrafish brain, this is a mouse brain, that Georg might talk about, and it has the same brain regions, the same neurons, the same neurotransmitter types. So the individual elements that make up this tiny brain are in principle the same as that in a mouse brain, because both of these animals are vertebrates, and therefore their function probably relates a lot also to humans and to human conditions. And with that, I'll hand it over to Georg, who will bring this whole presentation hopefully to a glorious end. Thank you.
More imminent than glorious, I hope. Very good. Yeah, just with the black line. Yeah, it's very good. Good. Perfect. Thank you. Very good. Yes. So, what I would like to tell you about is why I think this matters. right? Both Florian and Tom have very nicely shown that our senses are fooled by everyday experience. Why do I think this matters? There isn't consensus in neuroscience yet, and a lot of what I will tell you isn't accepted fact, but I would like to tell you about why I think it's interesting and where this could go. And first, I would like to point out this is more profound than just seeing colors or shapes. Right? The idea is that perception is controlled hallucination. Everything you see, hear, feel, think about the world is always a mixture of evidence you have from the world and your internally generated expectations. Right? You all have blind spots in your eyes right in the middle of your field of view. You do not see them. Your brain fills that in. Your voice sounds different when you hear it on tape. But when you listen to yourself or hear your voice on tape, I at least think, this is not me. This sounds weird. When I speak, my expectations of what it should sound like interfere with what I hear. When I sing, some people know how to sing, I don't. When I sing, I think I'm reasonably accurate. Right? Others tell me that's not the case. And I assume they're right. And the issue is likely that when you sing yourself, what you hear is a mixture of what you want to hear and what you actually hear. Right? People we know very well, right? They can come with a new haircut or new glasses and you don't see it. Right? It's things that happen right in front of your eyes and you just miss it. Right? You don't see them, and the things you see are the things you expect to see. When you think about why you do things, this is one that is very hard to demonstrate in normal people, in normal humans. But even your reflection on why you think you did certain things are very likely, very strongly influenced by why you think you should have done them, not why you actually did them. This can be shown in patients that are split brain. I will not go into the details, but roughly the experiment goes as follows. So these are patients where for medical reasons you separate the two cortical hemispheres. Now the left and right hemisphere can no longer directly talk to each other. Important to know is that only one of the hemispheres can speak, the other one cannot. And you can send information to only one hemisphere via the visual field. The right part of your brain controls the left part of your body, the left part of the brain controls the right part of the body. And now you can do an experiment where you basically fool the subject into doing something that the subject doesn't know why he, in this case, in this famous case I will tell you about, did it. So it's a patient that has this corpus callosum dissection, this split of the brain, and now you show the information. You give two pieces of information to the two separate brains. One is music. The hemisphere that can speak sees music. You do this by putting a word on the right side of the visual field. That only goes to the left part of the brain. And the other hemisphere sees bell. The right hemisphere that controls the left hand sees bell. And now you show 4 images to the patient and ask the patient to point at the image that best corresponds to the word with the left hand. Right hemisphere saw bell. One of the images is bell. All 3 other images are a guitar, a singer, something else to do with music. The patient obviously points at bell with the left hand, right? But only the left hemisphere can speak. And now you ask the patient, why did you point at bell, right? And there's an obvious problem, and the patient realizes that all the other 3 images are much closer to music, which is what the left hemisphere saw. And the answer the patient gives is, well, bells, music. Last time I heard music was the bells banging away outside. It's a complete lie. Right? It's completely made up. The patient in this moment made up this nonsensical story, but he's not aware of it. Right? He believes that is the reason why the hand points at the bell. Right? That the left hemisphere is not even aware of the fact that it has no control of what the hand is doing. Right? And obviously, unless one has a corpus callosum dissection, it's very hard to very hard to demonstrate that this is what's happening when we think about why we do things, right? But keep this in mind next time you get angry and then post hoc justify why you may have gotten angry. It's less of a problem when you're happy, of course. Then it doesn't really matter. Very good. So this very likely goes far beyond just funny perceptions of color, but to our understanding of how we interpret the actions of other people. Right? Does this person like me? Does this person not like me? Social dynamics heavily rely on your ability to make these inferences based on noisy and impartial data. Right? So why does this happen? Right? You would think, now we've heard all of this. Our internal models taint the way we perceive the world. Why does this happen? Right? Why would evolution come up with a system where we do not have access to reality as it is? Right? Why do we taint it? And I think, and this is speculation, but I think there's a relatively simple reason, and that is it allows us to predict the future. And this is usually the point where the audience goes, all right, I'll stop listening now, this guy is clearly crazy. Which is true, but not because the statement is wrong. You can predict the future, and I can prove that to you, or what I mean with this. I hope that's somewhat visible. Sorry, if you cannot see, sorry, this is the wrong image. This is the first one. So, well, you already see where this is going. The question is, what's happening next? Right? And we are so good at this that we would not even think of this as being able to predict the future. Right? At any given moment, you know exactly what's going to happen next. Right? You know now, and obviously, right? You can even imagine what this water running down my back here. This is how we do scientific arguments. Right? I lost this one. You can imagine how this feels like, right? You immediately, this is probably the basis of empathy, right? That you can look at something happening to somebody else and know what it would feel like, right? So this ability to predict and empathize and feel what somebody else is feeling is very helpful when you're trying to navigate the world, right? Predicting the future is essential to the point that you don't step out in front of an oncoming car when you cross the road, because you know 2 seconds from now, that car is going to be here. We are extremely good at this, right? Predicting the future for the next 2 seconds. If I suddenly disappear right now, right, you'd be very surprised because you've learned people don't suddenly disappear. Right? If I walk off stage 5 minutes from now, you're not too surprised. Right? So, but we're good at predicting the imminent future, and it's absolutely essential. Right? So why is this relevant? Right? I work at the Biomedical Research Institute. Why does this matter for medicine? Right? And the question, and this is again speculation, is what happens if this mechanism goes wrong? Right? The mechanism, sorry for making these predictions, right? Figuring out what's going to happen next. What's the issue, right? How could this manifest? And we don't fully understand how this works, but at least our one theory that I like a lot is that imagine, right, you have to find a balance. Everything you perceive is now driven by sensory input and your expectations. But you do not want your internal model, your expectations, to override the sensory input, right? Because then likely what happens is the thing we call hallucination. Right? If you start seeing things that are no longer there, that other people do not see, we describe this as hallucination. Right? And the diseases that are one of the diseases that is relatively prevalent, that's associated with, amongst other things, things like hallucinations and schizophrenia. Right? And this is what we think is happening, at least in part, in patients that suffer from schizophrenia, is that their system that is responsible for generating these expectations is too active, too strongly active. Conversely, when this system doesn't work, what's the consequence of not being able to predict the future? Not being able to have expectations. And there we think that might be one of the phenotypes of a disease we describe as autism. Autistic patients often describe the world as surprising, right? Everything that happens to them, they find surprising, right? It's unexpected. And when the world is too surprising, that becomes scary, right? And interestingly, many autistic patients resort to insisting on very stereotyped environments, right? They do the same things over and over again. Everything needs to be in the same place. This insistence on sameness is likely a strategy, a coping strategy that the brain develops to deal with this lack of ability to predict the future. Right? So we think of these two diseases as extremes on this spectrum when this fine-tuned match isn't fit properly, right? If you're too strongly reliant on expectation, then your perception is very strongly driven by what you expect to see, feel, and hear, or when those expectations are too weak. Right? And the type of experiments we do is we basically ask this question, right? What distinguishes something that is externally driven from what is internally driven? What we do, I'll just show you this to actually show you some of the research we do. Very briefly, we use mice that play video games like this, because now we can control what they see. We think we understand what they expect to see. We record neural activity in the brains of these mice, and then test the effect of drugs that we know work in these diseases, right? Antipsychotics, for example. Try to understand what is the mechanism of an antipsychotic in cortex, which we think underlies, at least in humans, these conscious perceptions, right? What happens, what is the circuitry in cortex that is responsible or is primarily affected by these antipsychotic compounds? And what happens when you give a mouse one of these drugs? What changes about the cortex, and how do we need to treat? How could we go about treating these diseases where this match between expectation and sensory input is no longer in balance? Thank you very much.
Okay, so I'd like to thank our speakers. I think it was very entertaining and interesting, and they tried to keep it less specialized so that you understand a little bit of the background of brain research. So the plan now is that we engage into a discussion about your curiosities about the brain. So you should be the ones asking the questions. There are 2 microphones in the room. You can just raise your hand and you will get the microphone until, yeah.
Do you hear me? Yes. Okay, what's your opinion about Deja Vu? I just had one while you talk. Nice.
Should I? Yes. To be honest, I'm not entirely sure I have anything interesting to say on Deja Vu. What we know is it's not understood. So what happens in these moments when you think you've seen something? But I mean, it is an essential feature of brains to understand when they recognize something. And how this works isn't entirely clear. If I give you a new word that you've never seen, you can instantly recognize that you've never seen this before. A scenery. I mean, whether it's actually true or not is a separate question, but you very quickly come to a conclusion that you've never seen something. How this works is absolutely not understood, because if you try to implement this in a computer algorithm, what you have to do is serially search, right? You go through in your entire dictionary of words or all the images you've ever seen to test whether you've seen this place or not, and by the time you reach the end, you conclude, no, it's not in my library. We can do this extremely rapidly, and my guess is that Deja vu is when this process fails, when something is slightly amiss with this process of figuring out whether we've already seen this or not.
Hello. So a very interesting detail I thought I could just throw in and tie all the presentations together. The mirror illusion that was shown, I think, in the first or second presentation, the spinning mask. It's a very interesting fact that patients with schizophrenia are actually more immune to these illusions. So they are less susceptible to the illusions. They correct better these these tricks that everyone should be doing, which is a very interesting thing because usually schizophrenia is known by a lot of people as the disease where you see things that aren't there. But on these visual tasks, they usually perform objectively better than control patients, which is a very interesting question. If we need a sort of baseline error to perception, if we need a little bit of uncertainty to not, I'm gonna just trail off.
Yeah, so the question, I think, if I may repeat, is that this mask illusion that you all fell victim to, is you could not help seeing a face that comes around, is indeed there are certain patients who are less prone to fall into this category, who can actually see it as a rotating mask. That was the comment, yes? And the question is, can we say anything about that? And I think Georg already has alluded to that. Is if this ability to predict is compromised, or even if it's in overdrive, then let's say if the ability to predict is compromised, then your brain will not insist that the next thing you'll see is another face that comes around, but it just takes the current image at face value and knows it's a hollow mask, right, when it's coming around, and is then less inclined to fall into this next state when the face comes around, because the prediction about the face is just less well developed. So the other case would be if you predicting things too well, then what also might happen is that the brain very quickly now overinterprets the incoming sensory information and falls into another thing, what's called an attractor state, or a perceptual attractor state, that now the maskness, the hollowness of the face now gets very, very quickly integrated into the model, and it will just keep running with that. So, I think one thing that's never been tried is use psychedelic drugs, and see whether if you're tripping, right, if you get high on LSD, whether then these illusions might take a very different form. That's an experiment that could be done. None of you should be doing that, right? I'm just saying, yes, I'm not telling you should do that, but maybe under control.
It's been done. Yes, no need to repeat. I assume the question was, it's slightly counterintuitive that schizophrenics would not fall into the illusion, and that is absolutely true. I mean, I glanced over this rather superficially. It's not entirely clear whether it's really just a single-dimensional spectrum. It's, for example, schizophrenics often tend to have auditory illusions and much less visual illusions. And why you have these asymmetries between the senses, as far as I understand, is absolutely not clear. It is possible that this is regional, that these defects are regional and affect part of the brain, and the rest then compensates in the other direction. That would be one possible explanation, but it's absolutely not clear. I mean, it's an excellent point.
Okay, so I have a question for Professor Florian Engert. So had you thought about studying neural circuits on organoids, or maybe another animal model instead of zebrafish?
So the question is, why fish and why not mouse?
Exactly.
Or
why not something more reliable, like brain organoids?
Oh, brain organoids.
Yes.
So I've started working on slices and organoids, and I think the problem with organoids is that they don't behave. And I'm not even saying this frivolously. I'm saying it because what we haven't managed to do yet is regenerate in an organoid the structure that's even capable of generating activity patterns that would allow an output, a robot, to control the world. So I've been involved in research that tried to interface organoids with robots, with machines, and ask how well can this organoid actually control the apparatus, and they totally suck at that, right? So I think that's one of the reasons. It's impossible to actually study the circuits that matter in organoids, because they really are totally incapable of producing behavior. What they are capable of now more and more is representing the world. You can use it like a feed-forward deep network, and I think there's some progress in organoids that you can encode stimulations. But as you've probably learned today, I hope the three of us have convinced you that a large aspect of the brain is not simply encoding information and representing it, but turning it around into a controlled feedback loop to now control behavior. And this is something that organoids cannot do. I'm not sure whether the question is also related, why not go up and work in mice? So what's the advantage of a fish versus a mouse? And I think there, the real superpower of the fish is that allows you to look at the whole brain. I'm not sure this is really coming through, but a mouse brain has 70 million neurons, and at most we can record at the same time from a few thousand, which is already a huge breakthrough. Yes, but it doesn't even get close to the whole circuitry. Not even close, right? Not even 1%. Yes, a fish brain, larval zebrafish brain, has only 100,000 neurons altogether, and these high-powered microscopes allow us now to look at the entire activity of the animal. And I think that for the first time gives you now the ability to look at distributed neural processing. So if there's several areas of the brain and several neural subpopulations that are critically involved, we can at least see them. We won't miss them. And I think that is the real power, that we can get the complete picture in an experiment. Is that making sense? Yes, sir.
I would have a question to Dr. Keller. You said that we have this indispensable capability of seeing in the future 2 seconds. I would like to ask if domestic animals like cats and dogs have this capability also.
Can you repeat the question, please?
I was asking Dr. Keller if he told us that we have this capability of seeing 2 seconds in the future. 2 seconds, not more. And I asked if domestic animals like cats or dogs have this same capability.
Yes. Yes. So yes, I mean, don't remember the 2 seconds. The point was that, you know, very close times we're good at, very long times we're typically not very good at. Yes, so with respect to animals, I mean, we study this in mice, and we know mice can do it. So to the best of my knowledge, it hasn't been done in dogs or looked at, but my guess would be that absolutely anything that has a brain can do this.
Yeah, so Georg's lab and other labs too, including ours, have done experiments where you can train mice to expect to see certain stimuli in the future, and then you can occasionally remove that stimulus when the animal expects it, and then there is an error signal, so there's a lot of activity associated with not showing anything or replacing a given stimulus with a different stimulus. And the kind of large parts of the brain or many different regions respond to that difference, indicating that they indeed have an idea of what will happen in the future. I don't think many people have tested how far ahead do you predict this, but I think Professor Engert has the answer.
He does indeed. He does indeed. So first of all, I think it's an excellent question, and I think it again drives home the point that the brain is projecting forward into the future. And all brains do it, not just domestic animals, any animal, I would say. It's almost the definition of an animal that it can do that, so that it can predict and try to control the future. Now the question, how long, this is the 2 seconds, is something that we don't know. Yes, and it's difficult, but it has something to do with time constants. Yes, is the time constants of the dynamical system that is the brain. And I have a novel theory that I'm going to share with you now, and that has to do with the question of why an elephant has a bigger brain than a mouse. And it's not just a little bit bigger, it's 1,000 times bigger in terms of the number of neurons. So an elephant has roughly 70 billion neurons, a mouse has 70 million. So it's a 1,000-fold difference, which is costly. A lot of neurons cost a lot of energy, yes?
It's even bigger than your brain.
It's certainly bigger than my brain, yes, which is not that difficult, yes. But so why is that? And then people say elephants are clearly much smarter than mice. So then I say, okay, then let's not take an elephant and a mouse. Let's take a hippopotamus or a rhino. I don't know what you think is the dumbest big animal. And we compare it to a marmoset monkey. Yeah, these tiny monkeys that are clearly very clever. And it's the same relationship. A hippo has a much bigger brain than a marmoset. And you wonder why. And here's the theory. Yeah. I think in order to generate these long time constants, which means that they project, I think a hippo or an elephant projects forward into the future by several seconds. And the reason why they have to do that is because their bodies move much slower. They can't move quickly because they're too heavy, and the force of the muscles does not scale with the increase in size. That problem, when the size increases more than the force of your muscles, that's also something that I and Georg experience as well, right? This problem when, when you get too heavy for your strength. But in the case of a hippo or an elephant, this results in everything moving a lot slower. What this means, I think, is that the response time, the projected time, needs to be much longer, because when they move, they have to predict now 5 or 6 seconds into the future before they put down a leg. And what should happen then? They also probably need to predict incoming objects much longer into the future because the reaction time is much slower. It also probably means that the working memory, they can remember then, because the whole feedback system is much slower, for many more seconds. A marmoset monkey would have much shorter attention span, would move much quicker. And the trick is that neurons operate on time constants of 20 milliseconds. If you want to generate long time constants with neurons that are short in time, one way to do that is just lump many of them together. So maybe an elephant has a bigger brain, not because it makes it smarter or it adds control or sensitivity, but it's one way evolution found of slowing shit down. Yes. This is very disappointing to theorists, because they believe that there is something complicated and awesome about having so many neurons. My theory, it's just a low-pass filter. It's just one trick that evolution found of slowing things down, and thereby, and that's a long answer to your question, allowing animals to predict further into the future and maybe also remember longer, slightly longer from the past. Okay, Yannick here thinks that an elephant is faster than me. Yes, sir. I'm not sure. I think I can totally take an elephant. Yes, just bring it on. No, but I think actually a related question to that is, how does the maximum running speed of any animal relate to body length? And if you work through the physics, what you realize is that the speed in which an animal can run depends on the stride length, which is the length constant of the animal, but it also depends on how quickly they can move their legs, right? What's the update frequency? And that turns out to be so the stride length goes down linearly, the longer you are, the bigger you are, but the frequency goes up linearly, decreases. And you can convince yourself, if you ever watch a big dog and a small dog walking next to each other, right? They have roughly the same speed. The big dog moves slowly, the small dog moves very quickly. So what this predicts is that the speed in which an animal can run is completely independent of the body length. And if you don't believe that, so I would argue that a mouse can actually match an elephant in any race, yes, sir, if it's the appropriate mouse. And those of you who don't believe that, ever watch squirrels and dogs and ask who's faster, or ever watch timber wolves, big timber wolves and small rabbits, and ask who's faster, and it's usually a match. So the answer to the question is running speed does not depend on body length. Yes, it's a constant, and the reason is because the longer stride lengths that you have is compensated for by the increased time constant. And the fact with the running speed and it's not correlated to body length is a fact. That's not even something, that's not shit I'm making up. I often make shit up, but in this particular case, this is actually a truth. Is the fastest animal, land animal, is a cheetah, which is right in the middle, right, of the distribution. And I think the second fastest animal is a hare, is a rabbit, which is relatively small. Anyway, so that's to that.
Hello, very nice and interesting information. I want to ask about the violence problem, because you show us something with a mouse, and I understand that there is something in the brain who can be damaged, and that's why the violence problem is in a human being, for example. And how can neuroscience fix this problem, if there are any answers, and how can we hope that it can be fixed for a long time? I'm sure.
Okay, I think you raise a really important question that we do not have the answer to that. Okay, so the question is, it's about violence. What do we know about how violence is represented in code in the brain, and can we do anything to fix it in those violent individuals? I think there might be two main reasons why individuals might be violent to others. One is they don't have a good sense of, if you want, some sort of, they have a pathology in interpreting how their actions affect others. So if they say they do this, they think they're not hurting anyone, but actually they are, or they even derive pleasure for it in some cases. The other case might be where the actual brain circuits that control aggressive behavior are dysregulated. They might be more active in certain states of the brain, maybe under alcohol or even in normal conditions. So the example I gave you here, so aggression, you know, okay, just brief introduction to the brain again that I didn't go into. You're going to have every one of us has a primitive part of the brain that is shared across all vertebrates, you know, frogs, fish, mammals, etc. And then there are bits that are kind of added on top over evolution that end up controlling parts of that primitive, if you want, lizard brain that was there, you know, 300, 400 years ago, even earlier, right? So these elemental circuits that are this primitive brain control the basic essence of aggression, or the motivation to perform aggressive acts. Sometimes that's triggered because you're attacked and you need to fight back, but other times it could be triggered without a particular stimulus. Or in humans, it might be somebody says something that upsets you and you become aggressive, right? But there are other, if you want, more cognitive parts of the brain that are enveloped that tend to exert so-called behavioral control or cognitive control, where they inhibit this kind of reflex that you naturally hit out at somebody and do that. So maybe it's that pathway that's also dysregulated. What scientists are doing right now, at least in terms of animal research, is trying to figure out these circuits, meaning which are the precise parts of the brain and which particular brain cells that go to different structures that control aggression. And I gave you an example of that in the mouse attacking the glove. But now also how these cognitive structures, say like your cortex, controls that primitive part of the brain. And that's the bit where I think we need more work on. If you have any, so we don't have an answer to that. It's because it's really tricky to study aggression in humans. It's easy to study aggression in mice because they tend to fight, right?
Yeah, I want to add something. So again, the answer is, if aggression goes wrong in humans, can we add anything to that from a neuroscience perspective? And I'll take that answer to share a story, not out of my own lab, out of a close colleague of mine, Catherine Dulac, at Harvard University. And she studied aggression in mice. And in particular, what she studied is what's called infanticide, meaning that male mice will kill babies. And it's a natural inclination, they do that. And the question is, can we understand the circuitry, the brain areas underneath it, and can we do anything to manipulate that? It's a very specific kind of aggression, but it's from a human perspective a horrible one. It's the males that kill babies. It is virgin males that do that. After they have mated, the virgin males, it takes them about 21 days to lose that drive to kill babies, so then they will become good fathers, and 21 days happens to be exactly the gestation period, so that's how long it takes until the baby is born. So this makes sense evolutionarily. Yes, sir. What Catherine Dulac did is she found the neurons that become active 21 days after mating, yes, by using tricks of the sorts that Tom was describing. Not only could she find these neurons in male mice, she could also now deliver one of these optogenetic probes specifically into these neurons, yes. So she was now generating male mice who were expressing this optogenetic indicator, it's not an indicator, this actuator, in these galanin neurons in the hypothalamus. And that's again a brain area that we all share with fish, with mice, with humans. So she made male mice that had this probe in the galanin neurons in the hypothalamus. She took virgin males, exposed them to mouse babies, and by the time they were about to attack and kill them, she switched on the light, the fiber, has activated these neurons, and these infanticidal male mice became good daddies. They started then grooming, putting them into nests, taking care of them. They were not as good as the mothers, yes, but still, they really turned into good daddies. Now, I need to say two things. Yes. This is very cutting-edge preliminary. You shouldn't get your hopes up, ladies. Yes. It's not as easy as that, yes. But so the time until we can implant an LED into the husband or the father, switch it on, and they start taking care of the kids, that time is still way in the future. But it still opens the possibility of at least thinking about how we could use this information that we get from basic research, yes, and at least start thinking about how we might be able to manipulate and cure, not cure, but dampen also human aggression.
So it's interesting that you mentioned this result, but do you think this is the level of understanding that you find satisfying, or do you think there's more than that? In a sense, it sounds like you can find some neurons in the brain that if you switch on or off, something is going to happen, and you can predict what's going to happen, but is there anything more than that, or is this all?
Florian making an excellent point is what can we learn, right, in terms of predictive coding and circuit motifs and computations in the brain. And Florian is right, we actually don't really advance our basic understanding of how the brain works. Yes. We can have identified now a group of neurons that is responsible for aggressive behavior, and we learn how to manipulate it. Yes. Florian is right, we have not really gained a deeper understanding of how the brain works, but we've gained some hope about being able to manipulate it and change a phenotype in a mouse or in a human. And I think that's an interesting point, because I think neuroscience research, in my view, has two completely separate goals. Yeah. One is to advance our basic knowledge, to make us more knowledgeable, more wise, more, give us more knowledge about the way how the world works, without any direct applicability or translational benefit, where the benefit really is an advancement of knowledge. And I think that in itself is a worthwhile endeavor. The other branch of neuroscience, or any biological science, is to actually help with the human condition. And in my experience, those two branches operate still largely independently. So big pharmaceutical research is not about basic understanding, it's developing something that works. Once it works, then the interest in how it works actually decays very quickly, is we have something that works. I want to make clear here that I think both of these branches are, of course, equally valuable. Yeah? So Catherine Dulac's research, right, is extremely valuable in giving us hope about actually helping people. It gives us little hope about gaining a deeper understanding of the brain. And that's sort of Florian's probing question, right? He knows that I'm not that excited about Catherine Dulac's work because it doesn't really help me understand the brain in any better way. Yeah? But I'm not saying it's useless, right? It's just useless for a particular question of getting a deeper understanding about predictive coding. Yes? It's immensely useful if you just want to have a hope, at least, or a pathway to helping with the human condition. So I'm a huge fan of both. Personally, I'm only engaged in the former, in the basic research, right? But I have enormous respect for the second. Does this answer your question, Florian? Yes.
It does. And perhaps I have a second comment, but maybe should I? So as you start to record more and more neurons and you start to have access to more and more information about the brain, at what point do you think you'll have enough understanding? What will make you happy to understand about the brain? And at what point do you think, aha, we have understood it?
When will you know?
And this is also related to something, does this work?
That was the question that I posed at the beginning on the slide for Florian to answer. Yeah. When will we know that we have understood the brain?
I got it. Yes, I have so the question is, when do we know we're there? Yeah? We have a goal to understand the brain. At what point would we have the answer? Are we happy with the answer? And for those of you who are really interested in that, I would like to direct you to a talk by Professor Markus Meister at Caltech. It's available on the web. Maybe the easiest way, if you're interested, write me an email. You can find me on the internet, and I'll send you the link. And what he does, he gives a talk which is called the standard model of the retina. The retina is the mini brain that all of you have in your eyes, that sort of has all the complexity and circuitry already encompassed. It has an input layer, that's the photoreceptors, an output layer, which is sort of the behavior of the retina of the eye, which is the ganglion cells. And in between, you have a relatively complex circuitry that sort of emulates a small brain. The number of neurons in this mini brain, called the retina, is 1 million. So this is 10 times more than a larval zebrafish brain. And in his talk, he gives an outline of what it means to understand this particular part of the brain, and even when you know you're done. And his answer is, you're done when you have a circuit model, when you can emulate your brain in the computer, and when your simulation generates the same kind of behavior that your animal will generate. So you make a circuit model of the brain, whatever you want to study, you run it, and you now do benchmark between your model and the real animals. And I would say if the similarity, if the information you can recreate goes above 80 or 90%, in Markus Meister's retina, it's about 80% of the information gets captured. But with respect to every spike in every neuron, right, so he can predict and simulate that, I think then you're done. So that's a sort of a somewhat hand-wavy, but it's at least an explicit recipe. So if you can generate a model that replicates the behavior of the real animal, I think then at least you're getting close. And you can actually quantify that, right, in terms of information captured of the output.
This also brings us to a question I always wanted to ask you. On a scale of 1 to 10, where do you think neuroscience is today in understanding the brain? You can use negative numbers if you want.
Well, I think the critical question is which brain. I think the C. elegans brain, the larval zebrafish's brain, yes, sir. If you use Markus Meister's metric, yeah, then I would say for a larval zebrafish, we are about at 5 or 6. Yeah. For the retina, I think we are at 8 or 9, yes, sir. For the mouse, I would say between 1 and 2, yes, sir. Well, the other question is, if you want to simulate the whole thing, the human brain, 1 or 2, yes, but the speed, the acceleration of how this number changes, how this dial changes, might actually move very, very quickly, especially if we can generalize.
Thank you. Maybe we should take a few more questions from the audience.
Yeah. Has it ever been considered for schizophrenic patients? For schizophrenic patients, like the treatment with electrotherapy? You said that you can shut down parts of the brain activity, and I wondered maybe that's a possibility for them.
So how much do interventions in humans? So the question is about electrotherapy in humans, how effective it is, what is the hope for the future? Is that roughly the question? Yeah. So there have been a few notable successes where you can put an electrode in humans who have some severe condition. Typical is, for example, Parkinson's disease, where we know a specific region in the brain, in that particular region that controls movement, or rather the initiation of movement. 80% of brain cells die, and they increasingly die off. And therefore, individuals find it very difficult to initiate movements. And they have other compensatory problems like tremor. If you lower an electrode into that structure that is depleted, or the target structure of those structures, then that rescues a lot of the problems. And it's a remarkable success, and it works in many individuals. Of course, the disease then continues to progress, and the individuals then might die for other reasons, but they can at least have reasonably normal movement generated. In other extreme cases, the cases of depression, as we know, that affects many of us. Some individuals are not, they don't benefit from any drugs they take or any cognitive behavioral therapy they take. And in those extreme cases, they have also done electrotherapy, where they stimulate particular parts of the brain, and that can also make them feel a little bit better, like the world won't end. So there are 2 successes. Now, as we learn, so the challenge for us now is, as we learn more about the brain, and we had a question about aggression, so there are these specific neurons that have a specific gene inside them called galanin, so we know exactly where they are in the brain. And if we can stimulate them or shut them down in particular ways, we can say, look, you as an individual are too aggressive. It would be good if you have this electrode put in there such that you can control your aggression, or indeed your partner control your aggression in case when you do. So that would be cool. The question is, there's a whole lot of ethics. There's a technological issue associated with that. Can we actually do it? But then there's a whole ethical layer on top. You know, historically, humans have allowed themselves to be experimented on. 100 years ago, which is not very long ago, people have voluntarily chosen to have frontal lobotomies. A frontal lobotomy is I wouldn't even call it a surgery, is a procedure where you take an ice pick, so handle and a pointy stick, that you go between your eyeball and the ridge of your nose, and you shove it in here, go into your brain and wiggle it around, and that destroys the front part of the brain, just like Phineas Gage, the railroad worker. And those people who are either have depression or are very, I don't know, have some usually social problems, many people have volunteered to have this surgery done on themselves between 1925 and 1940. In fact, several Hollywood stars have done it famously, much to their, so I don't know if there's going to be a brave new world where once the technology becomes safe, and that's the key bit, once we know there's no problems with having that kind of technology in your brain, whether you can then help yourself in the certain deficits that you might exhibit, or indeed, then if that technology is available, would you be able to even augment your performance, your ability to do certain things? I don't know whether that time will come. If the technology arises, I'm sure there'll be some legal way of making it happen, especially if it benefits the society in some way. And how far it will go, I don't know. And we can maybe discuss that, whether it should be allowed or not, right? Any other?
Hi. Perfect. This was really interesting. Thank you for your opinion. My question is kind of more a broad question. You can be as specific or broad in your answer of it as you would like. You spoke a little bit about schizophrenia and then also about autism and that being on two spectrums of possibly being able to, more or less well predict the future. Oncology 70 years ago was a very, confined field in the sense that we didn't really understand the causality of the cancer itself, right? But as brain in a dish technology, or brain in a dish, cancer in a dish technology kind of evolved, we were able to better, I guess, understand and then also combat these cancers. Notoriously, if you look at schizophrenia in a hospital or in a clinical setting, you have very different, clinical pictures of a disease that is classified or diagnosed as one sort of thing. In your opinion, are we looking at this the right way, or should we see autism and schizophrenia all on one spectrum of being able to predict the future more or less well and diagnose off of that? What are your opinions, mechanistically speaking, if we look at diagnosis in that picture? What are bigger and smaller mechanisms? Do we even know mechanisms that function in this sort of sense of being able to predict the future and being able to not predict the future? And also ethically, because we were talking about organoids, granting them at least, not agency obviously, but granting them the ability to control a robot with limited capacity is almost creating a soul religiously, that's mind-blowing. But ethically, how can we look at creating a soul in a dish, and how can we, because this is kind of the future of neuroscience, this is where we need to go with this, but how can we ethically kind of combine our view of the world with something that, at least personally, I feel needs to be done? I'm sorry this was very broad, but take it as you like.
Maybe with respect to the first of your question related to schizophrenia, yes, you have. It is probably important to keep in mind that all of these explanations of how it could work or what could underlie these things are, to my mind, motivations for why we should do the research. Right? It's a bit like saying you know, a car has an engine and something's wrong with it, but if you want to fix it, you need to understand how it works. So the thing where, I mean, most of our research is not about coming up with analogies and hypotheses about how these predictions might be impaired in schizophrenia, but in trying to understand what the circuitry is. And because we think eventually this will be relevant, understanding how this circuitry that generates these expectations, your predictions that you make, will be centrally affected in schizophrenia. These are things we're testing, just very broadly, but most of our efforts are going into what the circuitry is that is underlying these. So it's, you know, it's more of a motivation for why to do the basic research, and not a diagnostically relevant or even clinically relevant interpretation of what is going on.
I'll take the soul question. Yes, I think that's, let me unpack this a little bit. So the question is, what are the implications or the ethical considerations if we can generate a creature that has agency, and what's our responsibility for that creature that we've generated? It's almost like a Frankensteinian conundrum here. And I would say there's sort of two separate issues here. One is the issue of humans create something, and this is this arrogance with respect to God, right? That's one issue, right? Should we be allowed to create creatures? And I would say we've been doing this for the last 2,000 or 3,000 years by selectively breeding domestic animals, right? We're actually shaping brains, not explicitly through genetic or neuroscience manipulations, by selective breeding. And I think that is not really an issue, I would say, because I think there's even in the Bible, there is a command, right? Mach dir die Kreaturen untertan, yes, dominate the creatures. So I think that's not really a deep religious problem, is are we allowed to interfere with the generation of animals or not? I think by now everyone agrees that we probably can do that. So and whether it's an organoid or a chicken or a pug, right? I mean, these dogs, they really don't look like wolves anymore. I would say that's all on the same spectrum. So that's one. So I would not say that's really an issue. The other issue that goes also into the organoids, at what level through on the ladder of complexity should we start worrying about inflicting pain on a creature with a soul? At what level? Go from a fly? I would say an organoid currently is still below a housefly. Then we have a larval zebrafish, then we have a mouse, then we have a dog, then we have a monkey, and then we have a human. So there's a ladder of complexity. And my personal opinion is that the concern, so how much should we worry about suffering in all of these? And I think the main point, my point, is this is not binary. It's not either or, but it's an analog scale. Yes. The more we move upward on this ladder, the more we should worry. So we should worry most about humans, and we should worry least about organoids, for the same reason that we worry very little about houseflies or mosquitoes, right? We worry a little bit more about the fish, yes, than about the mouse and so forth. And I think that, to me, at least makes sense, yes. It's an analog scale, it's not binary, yes. And the question, at what point a soul enters the creature, whether it's a fly, an organoid, or an iPhone, I think I remain agnostic, yes. Maybe thinking about soul is not the right metric for that, right? But I think one correct metric is really how much should we worry about suffering in that animal. And again, I think it's a scale, and it's not binary. Does this make sense? Yes.
But I think if you were to ask a fly how much a fly should worry about a human, probably a fly would worry much less about a human than another fly or a bee.
You think a fly worries less about the human than the human about the fly?
Yes.
Well, yeah, that depends on the human and on the fly, right? Yeah, but that's probably true.
Hi. Hello. Here. So I have a. I should get up. Hi. Yeah. Okay, so you can see me. Nice. So since you all basically have argued that human actions can only be justified in hindsight, how can we then be more held morally responsible for our actions?
That's the free will question, right? Georg?
Well, I mean, this goes far beyond neuroscience, I think, because it's a legal question. But my interpretation would be that the legal system should protect society, and then it doesn't matter why. It's not a punitive legal system that we should aim for. I mean, this is not my scientific opinion. This is my personal opinion. But as soon as it's a protective legal system where you protect the rest of the society, then it doesn't matter. Right? If somebody is likely to do something again, it doesn't matter whether they have control over it or whether we think they have control over it or not.
Yeah, I can add to that. I think the question of responsibility, is it your fault? Yeah. Or maybe the excuse you're looking for is, Your Honor, it wasn't me, my brain made me do it. Yes, sir. And does that count? And that's complicated, because I think in some world you can say that's a valid excuse, but what Georg was saying, it actually doesn't matter. Yes, if you are likely to inflict damage on society, you should be removed such that you can't inflict damage anymore. Whether it's voluntary or whether you can't help it doesn't really matter. Yes. Like a rabid dog will be removed even if it can't help it. And there are certain human conditions where it can be traced back to a neurological problem. Tourette syndrome is one of the most famous ones, is where people uncontrollably will start cursing, yes, and lashing out. Yes, and there are other famous cases where you can diagnose a cancer, usually in the hypothalamus where aggression sits. Yes, and these diseases, these cancers can lead to uncontrollable aggression or even something like pedophilia. Yes, and what it can be used, I think it's accepted legal opinion now and neuropathological opinion that these people can't help themselves, yes, sir. So they shouldn't be punished, but they still need to be removed from society. And I think that's really, that's what Georg was leading to, right? It should not be about punishment and responsibility. It should be largely about making society safer. Yes. But I think a deeper question is that the ultimate responsibility is probably more like an illusion, right? It doesn't really exist, because ultimately, that's what we believe, it's always a biophysical neuronal network that's responsible for your actions. It's biophysics, yes. So there is really not a deeper point about arguing about responsibility. It's more like about the consequences of your actions need to be mitigated.
I'm curious to know about the link between dreams and the prediction the brain makes, and maybe its evolutionary function.
Sorry, what was the second?
The evolutionary function of it.
Well, dreams. The question is about dreams and the ability to predict, right? Okay, so here's my, maybe I'll go off on the deep end, and then Georg can rein me in and set this straight. My interpretation of how this all comes together is that this prediction engine that we've been talking about needs to be calibrated and tuned. So it needs to, this simulation, so what we've been saying is the brain simulates the world. It can simulate the world based on maybe some innate information that you're born with, but to some extent it needs to learn about how the world works. It needs to learn about these interactions. So it needs to build up this simulation engine. It needs to train it. And there's two ways you can train it. You can train it while online, but that's a problem, because then you have to train it with the same speed that you're actually behaving, because it's always about feedbacks from behavior. Or you could train the simulation engine offline. Yeah. So you first take the body offline somehow, and then you run the simulation engine and fine-tune it. I think that's what sleep is all about. That's why animals sleep. This is a deep, actually, conundrum. Why do we sleep half of our lifetime? What's the benefit evolutionarily? That was your other question, right? What's the evolutionary benefit of sleeping and dreaming? And that better is a good, you need a good answer for that, because it's immensely costly, yeah, to be defenseless for half of the, and it's dangerous to be that. So my idea is that what's happening during sleep is that the simulation engine gets updated and tuned. That makes two, if that's true, that makes two predictions. That makes one prediction that very young animals, like babies, should sleep a lot because they have a lot of updating to do. Yes, they learn about the relationships in the world during the day, and then they need to intake all of this information and update the simulation engine. Yes, the other prediction it makes, that if you don't sleep, yeah, if you prevent the simulation engine from running and calibrating itself, then it should start to drift and you should start making shit up, right? Like hallucination, hearing voices, you should go crazy when you stop sleeping. And that's also something that we actually observe. So that's my interpretation, and again, Georg might rein me in again, is that we sleep in order to run the simulation engine, and dreaming is exactly a memory we have when we wake up of this simulation engine in action.
That was very nicely put.
Oh, okay.
Scientists have found that during sleep there is a process of what's called replay, where some of the brain cells that were active during the animal's experience of the world, let's say it had to navigate some new environment, are replayed in a similar sequence, but not identical sequence, and they're replayed many times. And the idea is that what you're doing is you're refining that one individual experience that you had, and you're trying to make it more generalizable to similar such events if they were to happen in the future. So you're kind of making that, both, if you want memory, your model of the world more robust, such that it doesn't just depend on that one signal occurrence, but it generalizes a little bit around it. And there are specific mechanisms that have been proposed to do that, and they tend to happen during sleep.
If you have a microphone, just ask.
Yeah, sorry, I'll ask a very quick question. Where do these predictive computations occur? Are they strictly cortical, or could they happen in the retina, let's say? Thank you.
Yeah, so predictions happen in every structure in the brain that has been looked at. They certainly happen in the cortex, even though that's paradoxically least well studied. That's, for example, what Georg studies. Cerebellum, that's the small part of your brain in the back, that kind of predicts on a very far time scale how you should move your body and muscles given the input you're getting. That's been studied for several decades, and people know that. There is the dopamine system, which famously predicts rewards based on learned associations between a stimulus and a reward. That's a prediction machine, so that's well known. The retina predicts based on context and time. So, you know, if something is happening in this part of the visual field, the cells that are looking at the next part, nearby part, are, you know, changing how they respond accordingly depending on the context. So predictions are absolutely ever in the brain. But it's very strange, I mean, maybe we alluded to it indirectly, that the majority of brain researchers do not study predictions, which is very surprising given that, however, cognitive neuroscientists, and know, and we all know from our own experience, that primarily we generate predictions all the time.
And I think another answer to the question is are there localized regions that predict? I think it's not just, it happens everywhere. In many cases, also all these elements are involved. Yeah. So a prediction feedback loop can involve the thalamus, the cortex, the basal ganglia. So these loops, these feedback loops, right, often span many, many different areas. So it's not only that the individual areas do their part of predicting, often it's a distributed network that involves a large part of the brain, and they're all predicting together.
Hi. So my question is, how much of different experiences has a person under amnesia? And actually, based on the fact that the perceptions of the world are based on memories too, so how much do memories affect the perceptions of the world around us?
Yeah, hugely. This is
no,
okay.
yeah.
No. I just think you ask a really good question that is very difficult, right? So we know that, you know, amnesia, it can be induced in different ways, obviously. Well, you can have, so famous studies include Henry Mollison, or patient HM, who lost both of his hippocampuses. So these are the bits that are meant to be, you know, representing relatively recent episodic memories, okay? So there was a surgery, he had epilepsy, they took out those two structures, the two side hippocampuses, and he had no he had no recent episodic memory. He had short-term memory, so if there was a little star, he could draw the star, you know, that kind of thing. But he couldn't recall anything that happened recently. So if you know, you walk into a room, you tell him, hello, my name is Tom, and literally 10 seconds later, he will ask you, what's your name, right? So that kind of loss of memory. So let me just put it like this. I think your perception depends on this very short-term memory of what's just been happening over a few hundred milliseconds ago, because that helps you kind of combine all those things into some coherent percept. And I think that he had no trouble with. He had trouble storing sequences of events that happen in a coherent way that happen in the recent past.
Yeah. I think that it depends really what's the time constant again of your amnesia. And what Tom was saying, if it's a few minutes or 10 minutes or hours or maybe even years that you keep forgetting, then I think you become, it's harder for you to function in the world, yes, because you won't find your way home or whatever. But I don't think it really affects the way you feel or you perceive. However, I think if you shorten these amnesic time constants, if you lose the ability for working memory, there are a few relatively valid theories that would argue you would lose consciousness. Yes. You don't become conscious anymore, right? You lose the ability to perceive and be aware of the world. Yes, and that is interesting. That's sort of, and I'm tending to agree that might be one of the things you need for conscious perception is these short-term working memory.
Yeah, and that kind of links a little bit into Florian's new theory that we heard here for the first time about the time constants in the brain. So you need to keep things over relatively short time scales in mind. In this case, I'm talking about tens of milliseconds to hundreds of milliseconds time scale. So you can bind a bunch of things that might come at slightly different times, but help you kind of bind them together. What's interesting is that it seems like this very short memory that's necessary for your, if you want, conscious experience, is not localized to any part of the brain. It's distributed, right? And so that's very clear. Otherwise, there would be many more patients that would report such deficits of loss of. And the only time you really lose your conscious experience or perception is when you have massive brain damage, right? And then people are in a coma, for example.
Hi. So I have a broader question, and I hope I'm going to ask it right, so bear with me. So you talked a lot about how we're at level 1 or level 2 of how much we understand about the brain. And I wanted to know, what's your view of our current limitations in neuroscience? Is it a technological question? Is it the fact that we can't gather like enough data to put it in a massive computer and see what the brain does? Or is it rather how we conceptualize the brain and how we follow the idea of what we're looking for even? Right. And excellent. As a side question, sorry, since this wasn't enough, like what hope do we have of creating an AI there's anything like a human mind unless we first solve this puzzle?
Wait, yeah. Well, I'll do the first one. Is what's missing? Yeah. What's holding us back? And it's an excellent question because the common notion is that two things are holding us back. One is technology, that we can't record from enough neurons and not non-invasively enough. And the second, we don't have enough data. So I think they're both catastrophically wrong. And I can speak with authority because in the larval zebrafish, we have basically unlimited access technologically, and we can collect all the data we want. And what we figured out, it's basically useless. So it's not data that's holding us back, and it's not technology, I think. Yeah. What is it then? And I think it is actually the ability to phrase the question correctly. I think, in my view, it's like in physics, one should start with a big endeavor like that, within a model, a standard model, some intuition, and then we collect data in order to test the model or the intuitive working model that we have in mind. So I think what's holding us back currently is phrasing the question correctly. I think it should be informed, how we phrase the question should be informed by two things. First, what problem is your animal that you're interested in trying to solve, evolutionarily speaking? Yeah. How have these brains, for what purpose do they have evolved? Meaning, what's the explicit problem this animal is solving? And it's very different for a fish, for a mouse, for a monkey, or for a human. But that's the first thing you should identify. And then the next thing you should identify is how exactly are the animals solving this? What tricks are they using? In my experience, usually what cheap hacks are they using? What shortcuts are they using to solve this problem? It's usually almost disappointingly easy how they solve these tricks. And only then, once you have a good idea about these two things, should you look into the brain and look for the implementation of how it's being solved. Make sense? So I think what's missing is asking the precisely right question.
To the second half of the question, artificial intelligence, when will we be here? When will it be more intelligent than humans? It already is. It really depends on what you mean by more intelligent, right? Artificial intelligence plays better chess than us, better Go, drives cars more safely as of recently, and as of a month ago can explain jokes better than humans. Right. I mean, this is not a joke. The Google released a network that can explain. You can give it a joke and explains to you why it's funny better than I can.
Can it also tell jokes that are more funny than mine?
I haven't tested that, but so it really depends on what you mean by better at, right? Many things these networks are already better at, and it will gradually, right? We're getting better and better at designing and making them to solve tasks that where they exceed at human skill. Very good. But then should we?
Okay. So thank you very much. I just wanted to say thank you to everybody, not to ask you to go away, but just that we've scheduled it somehow ending at 6. But thank you very much, everybody. Thank you very much, your speakers and organizers, for the talk. And all of you for the contribution. It was really lovely. What we want to say before anybody exits the room is that we can stay here longer and still take questions. So the speakers have raised their hand. Please let us stay longer. So whoever wants to sit and still participate, they can stay after I finish saying goodbye to everybody that wants to leave at the particular hour. What I wanted to say in particular is thank you to our wonderful hosts here at the university. They gave us this wonderful room. And also to tell you that if you want to keep up with our meetings, like I said, we plan to have one in October that is similar to what we have here. We have the news page of the university. We have the Facebook page of Teens Neurotwin, which is the seminar series. And we have a website, which is neurotwin-seminars.com. I think I should have written them down, but of course you can find all of us whenever on email and ask more questions. We also have a newsletter that will advertise these things. So thank you everybody again, and whoever has to leave, have a great evening. And I will now pass the word back to our speakers.
Yeah, and I just want to say you've really been a great audience. We've enjoyed this a lot, and there were excellent questions. And again, we'd be happy to stick around. Maybe those of you who have questions can just come forward or give heads up. Anyway, thank you all.